Let's continue with chapter 12. So another technique that we're going to talk about, we call it page or polyacrylamide gel electrophoresis. If you remember, we talked about gel electrophoresis, that we use it in order to separate DNA. Polyacrylamide gel electrophoresis is pretty much the same, but we use it for separation of proteins. So, and the gel that we're gonna use, it's made of a different chemicals, so that they call it polyacrylamide instead of agarose. And also this technique we perform using a vertical gel instead of like horizontal that we talk about it in the gel electrophoresis technique previously. And so again, in this there are some wells that we can load our protein there, and then we're gonna use like electricity in order to separate and move the protein through the gel. And protein can be separated based on their size and charge as well. So since as you know, proteins, they are made of amino acids, and they can have like different charges, those amino acids. They're gonna use a chemical, they call it STS, in order to denature the protein, and they can unify the protein like negatively charged. So and it's gonna help that the protein gonna when we attach it to the positive and the negative electrodes, so the protein gonna move toward the positive electrode due to that negative charge that it has. And then again, we can run some ladder or marker, and then by comparing to the ladder and marker, we can estimate the size of the protein and those bonds that we get in that gel. So this is also a very useful technique for visualizing the protein and separation of the protein on a gel. All right, so the next technique, which is the very common and important technique that they use it commonly in a lot of different labs, like molecular labs, in microbiology labs, and so it's a very important technique that you know it as PCR or polymerase chain reaction. So this is a technique that helps to amplify a specific DNA sequence, so we can make like millions or billions of copies of that gene or the fragment, or a fragment of the DNA which we would like. So they use this technique in research, forensics, clinical lab, and so in order to amplify the DNA, so for sequencing, cloning, paternity testing, identification of the pathogens, so very important technique that we use. So if you remember, we talked about DNA replication previously. So PCR is like pretty much it's like a machine that by using different temperature gonna make millions or billions of copies of the DNA that we have. So it's gonna replicate the DNA. So there is a very important enzyme that we use in PCR that they call it Taq DNA polymerase. So this is a DNA polymerase that we use in order to amplify the DNA. And since we use like high temperature during the PCR, and you know DNA polymerase is an enzyme that can be denatured at high temperatures such as like 96 degrees Celsius, that we use it in one step of the PCR. So they found this enzyme actually in a bacteria that they call it Thermus aquaticus. So that's why they call it TAC. This name came from the name of the bacteria that they extracted the enzyme from, and this bacteria lives actually in the Yellowstone hot spring, and is very resistant to the high temperature, so it can tolerate the high temperature easily. So they extracted this, and now we use it from the bacteria, and now we use it in the PCR. PCR has three steps: denaturation, annealing, and extension. Imagine this is our double-stranded DNA. So the first step, we're going to increase the temperature to 95, 96 degrees Celsius. So it's going to help in order to unzip the DNA. So it's going to break down those hydrogen bonds between these nucleotides, or these double uh, bonds or double-stranded DNA that we have. Then we're going to decrease the temperature something around 50 degrees Celsius. So if you remember, primers, they were small RNA sequences that they match like to the genes of interest, or the part of the DNA that we want to do the amplification. So the primers during the annealing gonna anneal or attach to the complementary part, and we have two primers, forward and backward, and we can design those primers and purchase them. And then during the extension, so we're going to increase the temperature to 72 degrees Celsius. So this is the optimum temperature for Taq polymerase or DNA polymerase. During the extension, the DNA polymerase is going to add the new nucleotide, and it's going to extend the new strand and make like the complementary strand to each of the DNA strands. And then after one cycle from one DNA, we're gonna have two copies of the DNA, exactly the same genetic material. So now think about, we're gonna repeat this cycle 30-40 times. So usually after 2-3 hours we can make a lot of copies of the DNA of our interest. So I'm going to share with you uh, some videos. I highly recommend to watch the video, so it's going to give you a better idea about how the PCR works, and you can visualize this like different cycles and different steps in the PCR. All right, so the next technique that we're going to talk about, and it's very important in sequencing of the DNA. So it's very important to know what are those genetic information on our DNA, or in our pathogen, or our bacteria or viruses. So these are like the techniques that we use for sequencing and understanding the order of the nucleotides in our genetic materials. So before that, I'm going to talk about uh, a molecule that they call it like DNTP. So if you remember, DNTP, these are like those nucleotides that we needed in order to make the new strands of the DNA, like adenine, thymine, guanine, cytosine. So a scientist, his name was Sanger, in 1970, he used these DDNTP, which are like this, they have the same structure as those nucleotides, but just he did some modification that so they are missing the hydroxyl group at their uh, carbon in their sugar, one of the carbons in their sugar, and usually when they attach to the chain of the DNA that we are making or amplifying it, is gonna stop the amplification. So when they come to the when they attach to the DNA, so then the DNA amplification gonna stop, so it's gonna terminate by adding this DDNTP. So just giving you this information. So I want to talk about one of the like first techniques that we use in DNA sequencing. They call it Sanger chain termination. So by using this DDNTP, so these specific kind of nucleotides, so these scientists were able to sequence the DNA, and so we could like figure out what is the order of the nucleotide in the DNA or genomic DNA of that microorganism. So I'm not going to give you too much details about like how these techniques of sequencing work, but I want you to have like some ideas about like some of these techniques. Sanger is one of the like oldest and technique that we have in sequencing. It's a very accurate and precise technique, and they still use it especially for bigger fragments. But after that, a lot of other scientists, they work on new techniques for sequencing, so that in 2005, so a group of companies, they started a technique, they call it next generation sequencing, and based on the name of the companies, they gave them different names such as Illumina sequencing, Pyro sequencing, and 454 sequencing. So these are like different kinds of like companies that they could sequence in a very faster pace and in a very low cost uh, sequencers. So in a very short amount of time, we could get a lot of hundreds or thousands of sequences. So it made it much more uh, affordable, and it's much more easier for researchers and clinical studies and all those like labs in order to do the sequencing. So and they use like different techniques for their sequencing of the DNA, or one of the technique that they use it in 455 sequencing, or they call it pyro sequencing too, that the DNA they're gonna chop and cut the DNA into smaller fragments, such as like about 400 base pairs, and then they're gonna add some adapters to the DNA, and then each DNA fragment can uh, move or can be amplified by PCR using specific primers that they design to these adapters, and then based on that, so they can they have like a column that they're gonna run those like PCR fragments that they amplify it, and then based on imitation of some signals and light, they can understand that like what is the nucleotide, or what is the sequence of the nucleotides in that organism, so very quickly. So but and whenever you find a new microorganism, a new like sequencing of that bacteria or virus or fungus, we always need to submit those sequences to the gene bank. So this is like a universal uh, like a how is the gene bank or data information, that whoever finds something new, we always compare it with the data that we have in the gene bank. And if the bacteria or fungus or whatever that we discover, so we have to publish it there to make sure everybody has access to all the recent discovery. So actually find 11 different bacteria, so that I submitted and published them in the gene bank. A very interesting, yeah, especially in the discovery labs, that they always find new species, new like bacteria that you need to work on, or strains of the bacteria. So this is a case study. So I want you to read it, and then based on the techniques that we talk about, see if you can figure out that what technique can help in order to identify the potential infectious pathogen. So you have the answer at the end of the slides, but I want you to think about it, and there is not just one specific technique. There can be more than one. You can use different ways in order to figure out what is the infectious agent. All right, so now you learn a lot about different biotechnology methods in the previous sections. Now we're going to talk about like some definitions that you need to know. Some of them are repetitions, but I think it's a nice summary for you guys, that you can learn about all those important definitions in this part. The first one is genomics. So this is the study of the complete sets of the genes that you have in an organism. So all the genetic information, so when you study them, so we call it genomics. So it doesn't matter like the genes on the chromosome or on plasmid, so all the genetic information of an organism. Then we have transcriptomics. If you remember, transcription means making messenger RNA out of DNA. So transcriptomics is the study of the RNA or messenger RNA, that you only gonna focus on the genes expression. So you're gonna focus on the genes that they're gonna ended up to make proteins. So you're gonna focus on just the gene expression, for example, in a healthy patient versus the cancer cells. So you're gonna compare like the gene expression in different conditions. Then we have metagenomics. And we also have metatranscriptomics. So metagenomics and metatranscriptomics, they help in order to study the genes and gene expression for like a collection of multiple species. So you just don't focus on one species. So you're going to study a collection of the species, or even a collection of the microorganism. For example, imagine you want to study all the bacteria genetic information that we have on the skin, or all the fungi transcriptomics messenger RNA that we have like on our feet. So it's like a study of the whole genetic information or the messenger RNA on environmental samples without like culturing them. Then we have pharmacogenomics, which is the study of the genes that can affect the individual's responses to the drug. So this is a very useful technique because each person is different. So and they can individually study that how the using of these drugs or medicine can actually affect the genetic information or the genes on that person. Then we have proteomics. So as you can imagine, this is the study of proteins. So you're going to study the function and the structure of the proteins. Then we have metabolomics. So this is the study of the metabolites. So you're going to focus on the metabolic processes in order to understand more like metabolic or metabolites. Then we have bioinformatics, very, very useful technique actually these days because there are a lot of techniques that we get a lot of data. So like for example, sequencing. So the getting the data is one part of the research, but doing the analysis of those biological data using a lot of like softwares and computer work, this is the job of the bioinformatics, that they can actually do the analysis and understanding and interpretation of those data. Then we have classic and reverse genetics. Classic genetics, so you have like a mutation and then you want to see that, for example, this mutation change with characteristics or phenotype of that person. For example, in classic genetics, imagine you have a patient that shows cancer, then you want to see that, for example, which mutation or which changes in the genes causes like this problem or this disease. Then reverse genetics is opposite. So you have the gene. So for example, you do the mutation in that gene, and then you will see that what changes can happen in the organism. So it's like give you like some information like after you did some changes and mutations, for example, in a gene. So you will see that if you change this gene, so then what's gonna happen to that organism. So it's in a like a reverse studying of the genetics. All right, then we have genetic screening. If you remember, we talked about reporter genes. So these are the genes that we use it in order to identify, to see, like, for example, if there is any gene expression, if there is like, for example, they use the the fluorescence protein that they actually got it from the jellyfish, and then they will introduce these genes to the mouse, and then you easily can see that under the UV light if that gene expression in organism happen or not. So you can see it in this mouse, like two of them. They have this green fluorescence protein, and which is like a reporter gene, and they can monitor the gene expression in that organism just by using this technique, which is a very cool way to identify or visualize the gene expression in an organism. And we actually have this technique, the GFP, in the lab too, that you're gonna see like the bacteria that they have this gene, so they can see as a fluorescence under the UV light. Another common reporter genes that we talk about it, we call it LAC-Z, so that encodes beta galactosidase, and this enzyme can break down the X-gal in the media, and then it can change the color from white to blue in the bacteria. However, the bacteria that we want are the bacteria that, because of we clone and insert that DNA to the plasmid, so it interrupted the beta galactosidase gene. So these bacteria are not able to make this enzyme, and they cannot break down the X-gal, and they're gonna stay white. So the white bacteria or the white colonies are the ones that is our interest. The blue colonies, they don't have our insert, so that's why they can make the enzyme and they can break down the X-gal in the media. But the one that we are looking for are the white bacteria colonies. So this is another like a gene reporter that, or reporter genes that we used in the lab a lot. So these are some of the genetically engineering pharmaceutical products, and as you can see, for example, insulin, previously we have to get it from the pancreas, from pig or cow, and it was not actually very pure, so it could cause like allergic or immune responses that could be harmful to the patient. But these days, by just finding those genes and making recombinant DNA or other biotechnological techniques, so we can actually make a lot of products and use it. For example, HRR natriuretic peptide, so this is like a they use it for treatment of heart disease, high blood pressure, and kidney diseases, DNAs. So it's important for the treatment of the cystic fibrosis. So erythropoietin for treatment of the anemia and kidney diseases. Factor 8, which is the important factor in clotting the blood in hemophilia. Hepatitis B vaccine. This is another important vaccine that they can get it through the recombinant DNA production. Growth factor or growth hormones, so these are important, especially in some of the genetic diseases that they have growth hormone deficiency or Turner syndrome or Burns. Insulin, so as you can imagine, for the treatment of diabetes. And interferons, so these are important cascade of proteins that we're going to talk about it in your immune system. Tetrasinomycin, so we use it as antibiotics, and tissue plasminogens activators, so they use it for stroke and myocardial infarction or heart attack. So these are just some of the products that we can produce during the recombinant DNA production. So another technique, which is very important technique, and This is actually a natural process that happens in a lot of bacteria, and now we use this technique in the lab. So we learn it from the bacteria, and now we use it in the lab for gene silencing. They call it RNA interference technology. So imagine, if you remember, we have a DNA, then the DNA can transcribe and make messenger RNA. So if imagine we have, or we introduce a complementary uh, strand of RNA to our messenger RNA, that they call it antisense. So if this antisense matches or attached to the messenger RNA, so it's gonna block the messenger RNA. So when you block or interfere the messenger RNA, then the ribosomes, they cannot attach to the messenger RNA. So when there is no ribosomal attachment, there is no translation, so they cannot make or the protein anymore. So this is like what happens in the bacteria when especially they have like a viral infection. So like bacteriophages, if you remember. So this the bacteria, they produce this RNA interference, and they can block the RNA from the virus, and then the virus cannot do protein synthesis, making capsid and protein replication. So we use it in the lab. So there are different kinds of RNA interference. One, they call it siRNA, small interference RNA, and then we have miRNA or microRNA. So both of them, they are important in gene silencing. So they're going to suppress the genes. So siRNA, it usually can target the messenger RNA, and it's going to block the messenger RNA, and it's going to inhibit the translation, or it can even cause the breakage or cleavage in the messenger RNA. So, and miRNA, so they usually also can inhibit the translation of the messenger RNA and can cause breaking down or breaking the messenger RNA. So they have both potential therapeutic application for the disease treatment. So this is like they use it a lot in the research in order to stop the translation of the messenger RNA, or to break down the messenger RNA that we don't want to make protein for us. Okay, so the next topic is gene therapy, very hot topic. So we're using an adenovirus vector to do the treatment and cure of the certain genetic diseases. There are a lot of diseases that or defective genes that can cause like a problem for us. So the main goal of gene therapy is to modify or change those genes in order to do the treatment of these defective genes that can cause problem for us. So there are a lot of course, possibilities and potential in this in gene therapy for treatment of the illnesses, but unfortunately there are some issues and some like ethical problems that can cause. So for example, these adenoviruses, they can cause like trigger your immune system, that can cause like inflammation and can even cause to organ failure. And then these virus vectors, they may infect the other cells, so they may, instead of just targeting that specific gene, so they can maybe damage and target like different cells or different parts of the DNA and can cause cancer. So also modified viruses, they can reverse, so they can um, mm, like reverse their genetic information and can become like infectious again. And also the inserted gene can uh, become inactive, and then it may also even affect other important genes in our genome. So we may be like short term, we know like this gene modification can help the patient, but we don't know long term that if these changes in the genomic of that person or that patient can cause like for example tumor formation, or can cause cancer or other issues for that patient. So still very important techniques, very useful, but we still need to do more research and studies for more application and more like efficiency of this technique. So these are some ethical concerns for gene therapy. So, so there are some concerns, for example, which genetic traits are worthy of being corrected. There may be some people they misuse it or abuse it. So, for example, instead of just focusing on those genetic diseases, they focus on, for example, the your eye color, your skin color, your height. So also, should gene therapy be used for cosmetic reasons or to enhance the human abilities? Should genetic manipulation be used to impart the desirable traits to the unborn? So, is every entitled to gene therapy, or could the cost of the gene therapy create new forms of social inequality? So imagine that the people are rich, they have access to it, but the people that they don't have that economical capabilities, so they may not have access to that technique. So it's not fair to the social. And also, who should be responsible for regulating and policing these inappropriate use of the gene therapies? So we have to make sure we have strong regulation and policies for using gene therapy too. So I really like this video. So this is about CRISPR, very common technique that they use it for uh, gene therapy, and especially in sickle cell diseases and also other diseases they use it, and it was very successful. But as I mentioned, we still have to do more research, and in order to be able to use this technique more commonly and with more confidence in treatment of the diseases. So it's like a short video, but I highly recommend you to watch it. Thank you, and I will talk to you soon.